talk about the introduction, the article is related with the metabolomic studies in identification of metabolites that are associated with breast cancer. By identification of the metabolomic biomarkers, it will help in diagnostic of breast cancer patient since the metabolite that present in the breast cancer patient is different with the metabolite that present in normal healthy person. The objective of this study is to identify a set of serum metabolites that discriminate between healthy women and breast cancer patient. And the second one is to establish a common set of metabolic features as general metabolic cancer biomarker that discriminate cancer patients from healthy control.
In this study, the researchers analyzed serum metabolite profiles of breast cancer patients using high-resolution mass spectrometry. First, blood samples were collected from patients with breast, colorectal, head and neck, and lung cancers, along with healthy controls. After processing the samples, they used a commercial kit in combination with a high-resolution mass spectrometer and UHPLC system to profile 389 metabolites. To ensure the accuracy of the data, rigorous quality control steps were implemented throughout the process. After addressing missing data and correcting batch effects, 284 metabolites were selected for detailed analysis. Then, statistical tests were used to compare metabolite levels, identify patterns and similarities between the groups. Lastly, a support vector machine classifier was developed to distinguish breast cancer patients from healthy controls, which undergoing 500 iterations for accuracy. It is important to note that all procedures were approved ethically. In this study, which using high-resolution mass spectrometry, researchers identified distinct changes in the serum metabolite and lipid profiles of breast cancer patients, revealing a breast cancer signature. This included lower level of amino acids like glutamate and lysophosphatidyl-cholines and higher levels of acyl-carnitines. Additionally, a broader analysis discovered a 42-compound multi-cancer signature that differentiate cancer like colorectal, lung, and head and neck from healthy controls. Of these, 23 of these metabolites overlap with the breast cancer profile, highlighting common metabolic disruptions across various cancer types. The multi-cancer signature achieved an impressive AUC of 0.98 in breast cancer classification with minimal impact from factors like age and fasting. This demonstrates the potential of serum metabolomics for early cancer detection and precise classification.
In conclusion, this article successfully revealed the related serum metabolites that are associated with the breast cancer. The using of high-throughput metabolomic approach had revealed a set of serum metabolites and also components of hypothetical multi-cancer signature in the breast cancer patient. The presence of the set of serum metabolites and the hypothetical multi-cancer signature can differentiate between the breast cancer patient and the normal healthy woman. The second one is the identification of a common set of metabolic biomarkers in order to discriminate healthy control from cancer patient. The common sets of metabolic biomarkers, which also known as multi-cancer signature, is a general metabolic cancer biomarker that can be detected in various cancer, which means the detection of multi-cancer signature can be in various types of cancer, not just breast cancer. It is because it is a general metabolic biomarker.
Hi everyone, in this video, I'm going to demonstrate how to use the Human Metabolome database to explore a wide range of data related to human metabolism and metabolites. HMDB is a comprehensive resource that provides detailed information about metabolite found in the human body. It is an essential tool for researchers studying the biochemistry of human metabolism, as it contains data on small molecules, which is metabolite, that are produced by or used in metabolic pathway within the human body. First, open Google. In the search bar, type HMDB. Click on the first link, which is the Human Metabolome database. After opening the website, you can see 5 different tabs. In the Browse tab, you can browse your HMDB large collection of metabolites using different categories. Similarly, in the Search tab, you can find different searches for metabolites based on this provided option. There is a convenient search bar located at the top right side. Now you can simply search for your desired metabolome. As an example, type glutamic acid. Each metabolite record in HMDB has 10 different data sections covering identification, taxonomy, ontology, physical properties, and more. You can also access the section by scrolling down and view some of the other descripted section. You will find the common name, a detailed description, the structure, the synonym, the chemical formula, and the average molecular weight. Now, click on the Physical Properties tab. You will find detailed information about the physical characteristic of a specific metabolite. There is also information on whether the metabolite is a non-biomarker for certain disease. You can also find the details of enzymes that interact with the metabolite, whether as substrate, activator, or inhibitor. By navigating the HMDB website, you can explore many aspects in depth for any metabolome and understand its broader relevance to human health and disease.
The study utilizes high-resolution mass spectrometry or HRMS combined with the absolute IDQ P400 HR kit to analyze human serum samples and identify metabolic biomarkers for breast cancer detection. The key principles include: first, HRMS technology, which detects and measures metabolites with high accuracy and sensitivity by analyzing their mass-to-charge ratios. This provides detailed molecular insights. Second, quantitative metabolite analysis, ensuring consistent detection of critical metabolites like amino acids and lipids through an automated workflow. Third, comprehensive data processing, using advanced tools like Excalibur 4.1 and MetIDQ software to generate precise metabolite concentration matrices and correct batch effects. Lastly, statistical and pathway analysis, applying tests like Mann-Whitney-U and Kruskal-Wallis to identify significant differences in metabolite levels and link them to cancer-related biological pathways. This research benefits society in several ways. In healthcare, it enhances early cancer detection through non-invasive blood tests, leading to timely treatment and improved survival rates. Personalized medicine is also supported by enabling more accurate risk assessments and intervention strategies. For oncology research, these findings contribute to developing targeted diagnostic tools, refining cancer screening programs, and understanding metabolic changes in cancer progression. The pharmaceutical industry can leverage this insight to create diagnostic kits, develop therapeutic interventions, and explore new metabolic targets for cancer treatment.
Now, we are going to talk about the advantages and disadvantages part of this presentation. For the supporting evidence, first, consistency with amino acid and lipid metabolism alteration. The studies of decreased serum levels of amino acids such as glutamate, histidine, and phenylalanine and lysophosfatidylcholines in breast cancer patients aligned with previous research. For instance, earlier studies using quantitative mass spectrometry also reported reduced concentration of amino acids and changes in lipid profiles, including lysophosfatidylcholines in cancer patients. Secondly is increased glucose levels. The increased glucose levels observed in breast cancer patients in this study are consistent with prior evidence that high glucose levels are associated with cancer cell proliferation. Studies on the Warburg effect support this finding by linking cancer progression to altered glycolysis pathways. The third one is metabolic patterns across multiple cancers. The multi-cancer signature proposed in this study overlaps with metabolic changes observed in other cancers, such as reduced lysophosfatidylcholines and diglycerides, as reported in previous studies on colorectal and lung cancers. This alignment strengthens the hypothesis of shared metabolic markers across different malignancies. Move to the conflicting evidence that the author had considered. First, lipid profile variations. While this study reported reduced levels of lysophosfatidylcholines and diglycerides in breast cancer patients, some other studies found increased levels of this lipid in similar cohorts. For example, studies using NMR and LCMS reported opposite trends, particularly in Hispanic population. The authors attributed such discrepancies to differences in analytical platforms, population demographics, or study designs. Second, glucose levels in other reports. Though this study observed elevated glucose levels in breast cancer cases, some previous studies reported decreased glucose levels. This inconsistency may stem from methodological variations, differences in fasting status, or sample storage conditions. The last part is variability in biomarkers. The study noted that while amino acid reductions were consistently reported across multiple studies, the specific types of altered amino acids varied. For example, some earlier studies highlighted different amino acid changes, such as increased glutamate levels, contrary to findings here. So what is the limitation of the method chosen? A potential limitation of the methodology in this study is the reliance on high-resolution mass spectrometry for metabolic profiling without fully accounting for potential variability introduced by biological and pre-analytical confounding factors. For instance, differences in sample handling, storage duration, and fasting status of participants could influence metabolic concentration. While steps were taken to minimize the effect of certain variables such as age-matched cohorts and adjustment for batch effect, the lack of standardized conditions for factors like fasting and storage across different cohorts introduced potential bias. These inconsistencies could affect the reproducibility and generalizability of the findings, particularly metabolites highly sensitive to pre-analytical conditions. The authors have acknowledged the need for further validation under controlled conditions to strengthen the proposed multi-cancer signature.